You know, usually when we talk about a medical diagnosis, there is this expectation of like mechanical precision.
Right, like it's a binary thing.
Exactly. You break your arm, the x-ray shows a jagged white line, and the doctor just points at the film and says, well, there is the problem. It is clean, it's broken, or not broken.
It's definitely a comforting way to view medicine. I mean, we really want to believe that disease is always that distinct from healthy tissue.
But then you step into the world of oncology, specifically when you zoom into the landscape of the tumor microenvironment, and suddenly that diagnostic clarity just vanishes.
Oh, completely. It's a mess down there.
Right. We're looking at an ecosystem that is entirely murky, where the lines between, a good cell and a bad cell completely blur. And that biological fortress is exactly what we are dismantling today in this deep dive.
It is a massive topic.
It is. We are pulling some incredible new insights from a groundbreaking 2026 multi-omics review article by Yage Fu and colleagues. This was published in Cell Death and Disease, and they are exploring something called the CAF TAM axis.
Yes. The CAF TAM axis.
Okay, let's unpack this. I want you, the listener, to imagine for a moment that you were trying to storm a medieval castle. But the bricks of this castle's walls are constantly shifting, and the guards patrolling the ramparts are actually your own brainwashed allies.
It's a nightmare scenario for any attacking force.
Right. How do you even begin to break in? So today we're exposing the cellular city's corrupted architects and its rogue police force who build these walls to keep our immune system out.
To truly understand this fortress, we really have to start by meeting the builders. In the biological world, these are the cancer-associated fibroblasts, or CAFs for short.
Okay, CAFs.
Yeah. And to appreciate how insidious this is, you have to realize that in healthy tissue, fibroblasts are essentially peaceful maintenance workers.
Like construction guys filling in potholes.
Exactly. They wander around, they lay down the extracellular matrix, which is the structural scaffolding of our organs, right? And they help heal wounds. They are the good guys.
But in cancer, they get hijacked.
Yes. The tumor secretes distress signals that biologically brainwash these normal fibroblasts, forcing them into a corrupted state. And what this 2026 paper maps out so beautifully is that these hijacked workers don't just become one generic type of bad guy.
They specialize.
Highly specialized. They differentiate into entirely different subtypes.
Let's get into the specifics of this workforce, because the division of labor here is just wild. We have the propagandists, the bricklayers, and the double agents.
That's a great way to categorize them.
Let's start with the propagandists, the ICFs, or inflammatory CFs.
These are fascinating. ICFs are driven into existence by specific genetic alarm systems inside the cell. When the tumor signals hit the fibroblast, it triggers this intracellular domino effect.
A domino effect in the genes.
Exactly. Specifically the IL-1R1, JK STAT pathway, and the NF-kappa B pathway.
Okay, for anyone who isn't a geneticist, what does that actually mean for the cell? Like, what is it doing?
Think of those pathways as internal sirens that get jammed in the on position.
Oh, wow.
The signal reaches the cell's DNA and forces it to constantly manufacture and secrete inflammatory cytokines.
Cytokines being the chemical messengers.
Right, primarily these molecules called IL-6 and CXCL12. These molecules act like chemical propaganda.
The megaphones.
Yes, they flood the surrounding tissue to recruit other bad actors. And critically, they actively suppress the CD8 positive T cells.
And the T cells are our natural assassins.
Exactly. They are the ones that should be killing the tumor, but the ICFs just suppress them.
So the ICFs are sitting back from the front lines, just shouting into chemical megaphones to confuse the local immune system. But someone actually has to build the physical walls of this fortress.
Right, and that falls to the myCAFs, the myofibroblastic CAFs.
The bricklayers.
Unlike the ICFs, which are driven by inflammatory alarms, myCAFs are activated by a molecule called TGF-beta.
Okay, TGF-beta.
When TGF-beta binds to the cell, it acts as a genetic switch that turns on genes for contractile proteins like ACTA2. This literally gives the cell the physical ability to contract and pull the surrounding tissue tight.
So they are physically tightening the scaffolding.
Yes. They become heavy-duty bricklayers. They release enzymes like MMP11, which reshapes the environment, and they deposit massive amounts of dense collagen. They make the tissue physically hard.
Like concrete.
Exactly. And there's a specific detail in the paper we really have to highlight here. A subpopulation of these bricklayers, called LRRC15 positive myCAFs, build walls so dense that they actually block modern anti-PD-L1 immunotherapies.
Wait, I want to make sure I understand the mechanics of that. how does a physical wall block a targeted molecular therapy? I mean, aren't the drug molecules small enough to just slip through the cracks?
You'd think so, but it's actually an issue of physical pressure.
Pressure.
Yeah. By packing the tissue with so much collagen, these myCAFs increase the interstitial fluid pressure inside the tumor. They pack it so tight that the local blood vessels get crushed.
Oh, so the drug can't even get there.
Right. The drug physically cannot flow into the tumor bed. It just bounces off the high-pressure barrier.
That is highly optimized parasitic behavior. So we have propagandists confusing the immune system and bricklayers crushing the blood vessels.
And then there are the APTFs.
The antigen-presenting CFs, the double agents. The paper notes, they express MHC2I molecules like HLA-DRA and CD74. Now, I have to push back here.
Go ahead.
If a cell is an antigen-presenting cell, that means it's waving a molecular flag saying, hey, look, here's a piece of the tumor. Shouldn't that flag help the immune system recognize and attack the cancer?
That is the logical assumption, yeah. And it's exactly why they are so deceptive.
How so?
Well, in a normal immune response, presenting an antigen works because it's paired with co-stimulatory signals. It's like a green light telling the T cell to attack.
Right.
But these APSCAFs present the tumor antigens without the green light. They act as decoys.
Wow.
The T cells interact with them, get confused, and eventually they just become exhausted and shut down.
So they're double agents leading the immune system on a wild goose chase.
In most cases, yes. But, biology always has exceptions, which makes this field so incredibly complex.
Right, there's always a twist.
The paper highlights that in BRCA mutant breast cancer, a very specific subtype of these double agents, called S1000A4 positive ABCAFs, actually restrains tumor growth.
Wait, really?
Yeah. In that specific genetic context, their antigen presentation somehow triggers a productive immune response.
It's like a corrupted double agent who suddenly remembers their original allegiance and decides to help the patient.
It's a great way to put it.
And just to round out the construction crew, the source also details McCAFs, which are metabolic hubs driving glycolysis to feed the tumor energy, and VCAFs, which physically build the vascular blood vessels to keep the tumor oxygenated.
It is a complete, specialized municipal workforce.
To keep these main architects straight, I like to use the IMA mnemonic for you guys listening.
Oh, that's helpful.
Yeah, so I stands for inflammatory CAFs, the propagandists with the megaphones. M is for myofibroblastic CAFs, the bricklayers pouring the concrete. And A is for antigen-presenting CFs, the double agents waving fake flags, IMA.
IMA. I like that.
So just to do a quick mini recap for you before we move on, the tumor takes normal wound healing cells and splinters them into this diverse, highly organized labor union that chemically and physically remodels the environment to protect the cancer.
That is a perfect synthesis of the CAF landscape.
So here's a question for you, the listener. If you had a single molecular sniper rifle, which of these hijacked architects would you take out first? The megaphones, the bricklayers, or the decoys?
It's a tough choice. But, a wall doesn't defend itself.
Right.
If a rogue T cell manages to break through the collagen and somehow ignores the propaganda, who is doing the hand-to-hand combat to protect the tumor cells?
And that brings us to segment 2, the security force. The rogue police of the cellular city tumor-associated macrophages, or TAMs.
TAMs, right.
Now, earlier I mentioned the brainwashed allies on the ramparts. Macrophages are usually our most aggressive defenders. How do they get compromised?
Well, historically, medical textbooks taught a very simple, almost cartoonish binary model for macrophages.
The good cop, bad cop routine.
Exactly. M1 macrophages were the good guys that caused inflammation and ate pathogens. M2 macrophages were the bad guys in cancer, promoting tissue repair and tumor growth.
Right.
But this 2026 paper definitively declares that the old M1 versus M2 model is just, well, it's completely dead.
Right, because single-cell sequencing has blown that wide open. We aren't just dealing with two buckets anymore. We are dealing with a terrifying spectrum of specialized guards.
Yes, the TAMs are highly diverse.
Let's look at the worst of the worst, the SPP1 positive TAMs.
SPP1 stands for secreted phosphoprotein 1, which is also known as osteopontin. These macrophages are highly destructive.
The riot police.
Yeah, they secrete VEGF, which is a protein that forces new chaotic blood vessels to grow, supplying the tumor with nutrients. And more importantly, they release MMP9.
Which act like molecular scissors, right?
Exactly. MMP9 degrades the healthy surrounding matrix. It literally cuts away normal tissue to make room for the tumor to expand.
Wow.
And crucially, they physically bind to the CF architects we just talked about,
Yeah.
using something called the SPP1 CD44 axis. They basically lock arms with the builders to coordinate the expansion.
That is terrifying. Then you have the LAMs, the lipid-associated macrophages. If the SPP1 TAMs are the aggressive riot police, the LAMs are the corrupt cops taking bribes.
I love that analogy. LAMs are marked by a surface receptor called TREM2, and their defining characteristic is that they gorge themselves on lipids and cholesterol found in the tumor microenvironment.
But why does eating fat make them bad? Like, how does that actually help the tumor?
It comes down to metabolic reprogramming. When these LAMs gorge on cholesterol, it physically alters their internal state.
It changes them.
Yeah, they become so stuffed with lipids that their internal machinery just shifts. They stop producing the inflammatory chemicals needed to activate T cells, and instead they start secreting immunosuppressive molecules.
Oh, I see.
By indulging in that metabolic gluttony, they effectively put the local immune system to sleep.
That is fascinating. The tumor bribes them with cholesterol, and in return, they just turn a blind eye. Now, what happens if the tumor grows so fast that it outpaces its own blood supply and runs out of oxygen?
Then the tumor calls in the angio-TAMs.
Angio, meaning blood vessel.
Right. These are found deep in the hypoxic or oxygen-starved areas of the tumor core. The lack of oxygen triggers a genetic switch inside these macrophages called HIF1A.
HIF1A.
Yes, when HIF1A flips on, these macrophages start building emergency blood supply lines to keep the tumor from suffocating.
So we have matrix-busting macrophages, lipid-gorging macrophages, and emergency blood-building macrophages. But just like with our double-agent CAFs, there is a wild card in the police force.
There is, the C1q positive TAMs.
Yeah, the C1q ones.
What's fascinating here is that C1q is a protein that's part of the complement system, which normally helps clear out dead cells and microbes.
Okay.
C1q positive TAMs are highly phagocytic, meaning they love to physically eat other cells.
They're the eaters.
In most cancers, they use this ability to clean up the tumor's mess and suppress immunity. But surprisingly, the paper points out that in melanoma and lung cancer, the presence of C1q positive TAMs actually predicts a favorable response to immunotherapy.
Wait, a favorable response?
Yes.
Meaning that if a researcher is looking at this, they have to wonder if we have these massive eater cells just hanging around the tumor, could the right immunotherapy suddenly weaponize their appetite and turn them against the cancer cells?
Precisely. The diversity of TAMs means we can't just broadly wipe out all macrophages. We have to understand their specific roles.
Okay, let's pause for another mini recap for you. TAMs are highly specialized. You've got the matrix-busting SPP1 TAMs, the lipid-gorging TREM2 LAMs, and the unpredictable C1q TAMs.
Dot on.
So my question for you, the listener, is this. Knowing macrophages can physically eat other cells, how could we theoretically brainwash them to turn their appetites back on the tumor? Just something to think about.
It's the billion-dollar question in oncology right now.
So we know the builders, the CFs, we know the guards, the TAMs. But how do they coordinate? Because a city doesn't function if the construction workers and the police aren't talking to each other.
Right, they have to communicate.
And this is the absolute core of our source material, the CAF-TAM axis. They don't just exist in the same space, they are engaged in a constant bidirectional crosstalk. Let's break down the mechanics of that communication.
Well, first is chemical recruitment. CAFs that express a specific protein called FAP secrete that chemokine we mentioned earlier, CXCL12.
The propaganda megaphone.
Yes. This chemical acts exactly like a homing beacon. It floats out of the tumor into the bloodstream and binds to a receptor called CXCR4 on the surface of wandering, innocent white blood cells called monocytes.
It essentially kidnaps them off the street.
It does. The CXCL12 signal pulls these innocent monocytes into the tumor microenvironment and forces them to differentiate into those corrupt, fat-eating TREM2 positive LAMs we just talked about.
Unbelievable.
The architects are literally recruiting and brainwashing their own security force.
But the communication goes both ways, right? The security force also gives orders to the architects.
Right. Those aggressive SPP1 positive TAMs secrete their own cytokines, specifically IL-1A, IL-1B, and TGF-B1.
So a whole alphabet soup of chemicals?
Basically, yeah. Think of these as chemical work orders. These molecules wash over the FAP-positive CMFs, binding to their surface receptors and commanding their genetic machinery to mass-produce type 1 collagen, or COL1A1.
So the guards are actively commanding the builders to make the walls thicker.
Exactly. And the physical result of this teamwork is spatial topology.
Spatial topology, what does that look like?
If you look at this under a spatial transcriptomics map, which is basically a high-tech biological GPS, you see them forming a structure called the tumor immune barrier, or TIE.
BIB.
Yes. They form a literal physical ring at the outer edges of the tumor. These SAMs and TAMs are standing shoulder to shoulder, physically trapping our heroic T cells outside the fortress walls.
It's a literal shield wall. But if we connect this to the bigger picture, the most profound discovery in this paper is a phenomenon called macrophage myofibroblast transition, or MMT.
MMT. This is where it gets truly mind-bending.
Here's where it gets really interesting, folks. They aren't just working together, they are literally shape-shifting into each other.
Yes. The paper details how, under the influence of a signaling pathway called SMAD3, M2-like TAMs undergo an epigenetic transformation.
Okay, let's slow down. Epigenetics. That is how genes are turned on or off without changing the underlying DNA, right?
Correct. Under this SMAD3 influence, the macrophages stop expressing immune markers and literally transform into ACTA2 positive CAFs.
It is like a police officer taking off their uniform, putting on a hard hat, and realizing they now have access to a completely different set of tools to pour concrete.
That's exactly what's happening. The tumor is so desperate for infrastructure that it forces its police to become construction workers to accelerate the fortress build.
It is a level of cellular plasticity that completely changes how we view tumor biology.
It does. These aren't static cell types. They are fluid states dictated by the needs of the tumor.
Okay, time for a quick recap. CTAFs and TAMs communicate via CXCL12. They build physical barriers together, called the TIVE, and they can even undergo cellular transdifferentiation, MMT, to shapeshift.
Right.
So for you listening, if you were designing a therapy, would it be better to break their chemical walkie-talkies, the chemokines, or dissolve the physical wall they just built?
It's a difficult puzzle.
Which brings us to our final exploration, breaking the fortress. If you are an oncologist looking at this chaotic, shape-shifting, heavily guarded fortress, how do you lay siege to it?
Well, for decades, the strategy in cancer was to just nuke the site with systemic depletion.
Just kill everything.
Right. But depletion, just trying to kill all macrophages or all fibroblasts, often fails. It's wildly toxic to the patient because you need those cells for normal bodily function.
And the tumor just replaces them anyway, right?
Exactly. If you just kill them, the tumor's chemical beacons are still active, so it just recruits more innocent cells to replace them. The paradigm is now shifting from depletion to reprogramming.
Reprogramming the workers. Let's look at the specific clinical strategies they outline in the text, starting with the TAMs. If we can't kill them all, how do we neutralize them?
The paper highlights blocking the CSF1 to CSF1R survival pathway using a drug called pexidartinib.
Pexidartinib.
Yes. CSF1 is a chemical that macrophages rely on for basic survival in the tumor. By blocking its receptor, you essentially starve the corrupt macrophages out of the tumor without nuking the entire immune system.
It's much more targeted. And then there is the CD47 SRP alpha blockade using drugs like magrolimab. This mechanism is incredible.
It really is.
CD47 is a protein that tumors express on their surface, which literally broadcasts the chemical "don't eat me" signal to the macrophages.
A biological cloaking device.
Exactly. Magrolimab binds to that signal and puts a blindfold on the macrophage. It can't see the "don't eat me" sign anymore, and suddenly the macrophage's natural ravenous appetite turns back on, and it just devours the tumor.
It's brilliant. We also have CAR macrophages, where we take the patient's macrophages into a lab, genetically engineer them to act like biological terminators that solely hunt the tumor and put them back in.
Just hunting the cancer cells.
But we must also target the CFs. Because many of the worst CAFs express the FAP protein, scientists have developed FAP CAR T cells.
So training the body's T cells to specifically assassinate the corrupt architects.
Right. They also mention Syncon FAP DNA vaccines, which train the body to constantly hunt those FAP-positive cells.
And there's this wild technique called near-infrared photoimmunotherapy, or NIRPK.
Oh, that one is amazing.
They use specific antibodies bound to a light-sensitive dye. The antibodies attach to the CFs, a doctor shines near-infrared light on the tumor, and it specifically melts away the FAP-positive fibroblasts without harming the normal tissue right next to it.
It's incredibly precise. But perhaps the most elegant therapeutic approach is reprogramming the CFs back into a peaceful state.
Yes. The paper explains that using simple vitamin A, in the form of ATRA and vitamin D, can reprogram these cells. It absolutely amazes me that something as familiar as vitamin A and D can neurologically soothe these corrupted builders into laying down their tools.
It's almost hard to believe, right?
How exactly does a vitamin undo cancer brainwashing?
It comes down to the origin of the cells. In pancreatic cancer, for example, many CAFs originate from pancreatic stellate cells, or PSCs.
Okay, PSCs.
When these cells are healthy and quiet, they store massive amounts of retinoic acid, which is vitamin A, in little lipid droplets. The tumor forces them to dump their vitamin A stores, which physically alters their chromatin and makes them angry and active.
So they lose their vitamins and go crazy.
Pretty much. By flooding the system with ATRA, we artificially replenish their vitamin A stores. It chemically forces their chromatin back into a state of cellular slumber. They literally lay down their tools.
That is just wild to think about. So what does this all mean for the physical fortress? If we can't soothe them with vitamin A or assassinate them with, say, our T cells, we just dissolve the wall they built. The paper discusses an engineered enzyme called PEGPH20.
Right, PEGPH20. So the scouts pack the extracellular matrix with hyaluronic acid. Now, hyaluronic acid acts like a biological sponge. It absorbs massive amounts of water.
Which is what creates that intense physical pressure inside the tumor we talked about earlier.
Exactly. PEGPH20 acts like a molecular acid. It specifically melts the hyaluronic acid. When that sponge dissolves, the trapped water is released. The interstitial pressure suddenly drops to zero, and the chemotherapy that was previously bouncing off the wall can finally rush into the tumor core.
It's like opening the floodgates.
It changes the entire physical dynamic of the tumor.
So just a final quick recap. We are moving into an era where cancer treatment looks less like a bomb and more like a software update for your cells.
I love that phrasing.
We're turning off the don't eat me signal for TAMs, melting the matrix with enzymes, and using vitamins to pacify the CAFs. So my question for you listening, is this the end of traditional chemotherapy as we know it?
It's definitely a massive shift. If we synthesize everything we've mapped out today, the overarching lesson is that a tumor relies entirely on this complex, dynamic ecosystem.
It's not just a lump of rogue cells.
Exactly.
Yeah.
If an oncologist simply uses a drug to kill the mutating cancer cells, but leaves the corrupted CAFs and the TAMs intact and communicating, that fortress is just sitting there. It's waiting for a new rogue cell to take the throne. The infrastructure of the disease remains fully operational.
You have to dismantle the infrastructure. But here's a final provocative thought to leave you with, building on the paper's conclusion regarding the future of AI-guided modeling and 4D spatiotemporal dynamics.
The 4D aspect is the real frontier.
What if the future of oncology isn't just about selecting the right drug? What if it's about using artificial intelligence to map your specific tumor in real time, watching the CFs and TAMs physically move and communicate?
Tracking them as they shift.
Right. What if the AI waits for the exact millisecond? The CAF-TAM barrier shifts its topology and then launches a chronologically choreographed strike.
This raises an important question about the dimension of time and treatment. It shifts medicine from just spatial biology to temporal warfare, timing the collapse of the barrier perfectly.
Even crazier, what if we eventually realize we have to let the tumor build a specific temporary barrier just so we can use a highly targeted drug to collapse that exact barrier onto the tumor itself?
Using our own defenses against it.
Exactly. So the next time you think of cancer, ask yourself, are we really just fighting a disease? Or are we negotiating with and ultimately trying to outsmart a deeply corrupted, highly adaptable cellular city?
It's definitely the latter.
Keep questioning the underlying mechanics and keep diving deep into the science.